Okay, great. Thank you. So first of all, thank you, Edward, for this nice introduction. Great to see you again. And thank you, Edward and Catherine, for the invitation. I'm very happy to be here with you today to talk a little bit about the things I do here in Germany. Since 4.5 years ago, I am a junior group leader here at the Technical University of Berlin. I am the head of the Theoretical Structural Biology group. Can you hear me well, by the way? I hope you can. Edward, can you hear me? Okay.
Yes, I can hear you perfectly.
Okay. Okay, if you in the seminar room have a problem, just let me know. Yep. And then again, I am the head of the Theoretical Structural Biology group, and what my research group does is modeling and simulation of protein-ligand interactions in vitro and in vivo. So one of the things we do is to look at ligand binding kinetics. We basically explore kinetics by using physics-based methods like molecular dynamic simulations and enhanced sampling, and also using machine learning. And this will be the topic of our talk today. And the other thing that my group does is investigation of crowded cell-like environments. And for this, we use physics-based methods like molecular dynamic simulations or Brownian dynamic simulations. I'll not talk about it today, but you feel free to ask me questions if you have any. And then getting started with the seminar, first, the study of kinetics is very much related to optimization of protein-ligand interactions and protein-ligand binding. So to optimize drugs, in the past, it was very common to look at the thermodynamics of protein-ligand binding. And thermodynamics applies for equilibrium conditions. And here we have a simple scheme, the protein and the ligands here in the unbound states, and they form, if they bind to each other, they can form the bound state, the protein ligand complex. So in equilibrium conditions, we can, to characterize this interaction, we can either determine the Kd, the dissociation constant, or delta Gb, which is the binding free energy. And the Kd or the delta Gb are different ways of measuring the affinity of the ligand for the protein. So Kd, for instance, is given by this equation here, where P and L are the concentrations of the protein and ligand alone in equilibrium, and PL is the concentration of the protein ligand complex in equilibrium. And Kg is related to delta Gb by this equation here, where R is the gas constant and T is the temperature in Kelvin. But what I want to highlight to you today is not thermodynamics, but the kinetics of protein ligand binding. And kinetics, it applies not only to equilibrium conditions, but also to steady state conditions or to non-equilibrium systems. And kinetics is special because, again, it's a better group of descriptors for non-equilibrium conditions, and a very special class of non-equilibrium systems are us and our cells. So our cells are out of equilibrium. They're constantly taking glucose from the environment, converting it to CO2 and ATP. And if we are in equilibrium with the environment, we are dead. So we are non-equilibrium systems. And then in steady state conditions, we can talk about K-on, which is the rate of complex formation, and also about K-off, the rate of complex dissociation. And what I will highlight to you today is especially K-off. And sometimes instead of talking about K-off, we may refer to the residence time. And the residence time, or tau, is just the inverse of K-off. And this is a bit more difficult to investigate because K-on and K-off, different from the thermodynamic properties, they are not state functions, meaning that they depend on the path we use to move from one state to the other state, from the bound to unbound state and vice versa. And kinetics has a lot of nice applications in biotechnology and also in drug design. So talking a little bit about binding kinetics in drug design. So the idea of optimizing drugs by looking at the drug protein residence time was proposed in 2006 in this paper here at Nature Reviews Drug Discovery. And since then, a lot of groups have been working, doing experiments or simulations to characterize kinetics of protein drug binding and unbinding. And one of the things that motivates this is that kinetic rates, in many cases, are better correlated with in vivo efficacy of drugs. And this makes sense if you think about it, because the longer the tau, the longer the residence time. This means the drug spends more time bound to the protein. We have a longer physiological effect and also less of target binding. And here we have an example of this. So basically, this example comes from this paper from 2012. The researchers were looking at inhibitors of the adenosine receptor and measuring efficacy inside cells. So here on the y-axis, we have the efficacy here and here. And here in the x-axis, we have the inhibition constant, which is a thermodynamic property. And here we have the residence time. So we can see that we have a much higher coefficient of determination when we compare the efficacy to the residence times. And binding kinetics also has interesting applications in biotechnology and enzyme engineering. So first, according to this review here from 2019, more than 60% of enzymes have their active sites located in deep internal cavities. This is important for enzymes to protect the active sites from non-specific interactions, but this also brings some challenges for substrate binding and product unbinding. And this also means that the access tunnels can be modified and we can further optimize substrate binding and product release in enzymes. And one example of this comes from this paper from PNAS from 2014, where the researchers were investigating this enzyme called epoxide hydrolase that uses NGE as the substrate. So first, this group of researchers did some simulations to see substrate binding and product unbinding in the Y-type enzyme. And what they realized is that while substrate binding was not blocked, there were no barriers for this. For product unbinding, product unbinding was hindered by some bulky residues, particularly in positions 145, where there was a methionine, and in position 128, where there was a phenylalanine. So these two groups were making it harder for the product to be released. So based on the results from the simulations, these researchers designed mutations in these two positions to alanine to make way for the products to be released, and they measured the catalytic rate for the Y-type enzymes and for the mutants to alanine in these two positions. And what they realized, as expected from the simulations, is that while the catalytic rate for the Y-type was 0.6 per second, the catalytic rate was much higher in the two mutants in comparison to the Y-type. And this was according to the simulations that indicated that these two mutations would facilitate product release and make the enzyme more efficient in catalysis. And then I hope you're convinced that binding kinetics is a very nice topic. I find it very nice, but I'm biased because I work with this. So what we'll talk about today is basically the work that my research group has been doing in exploring binding kinetics in nickel iron hydrogenases. And if we have time in the end, I'll also talk a little bit about binding kinetics in toll-like receptors, which is more related to drug design. For most of this lecture now, we'll talk about nickel iron hydrogenases. So this is a very popular model system here in Berlin because there are a lot of groups interested in applying spectroscopy to understand catalysis in hydrogenases. And hydrogenases are also a model system to understand hydrogen, molecular hydrogen formation and cleavage. They basically catalyze this reaction here. And this is a basic scheme of a hydrogenase, the small subunit in green, the large subunit in pink. And then we have here the catalytic site where the formation or cleavage of the molecular hydrogen happens. And some hydrogenases, so hydrogenases, first of all, they have a very important biotechnological application here in Berlin because we can use them to produce molecular hydrogen, which is considered the fuel of the future because if we burn molecular hydrogen, we do not generate CO2, but we still have the energy. And hydrogenases are a way of generating molecular hydrogen, But some hydrogenases, they are limited and we cannot use them efficiently because some hydrogenases are inhibited by CO and O2, which are common gas molecules present in the atmosphere. And before we did simulations with this hydrogenase, so what the hydrogenase we investigate is particularly this hydrogenase from the sulfonylurea because this has reasonably high efficiency for molecular hydrogen generation. And other groups already started investigating this hydrogenase and also tried to see if it was possible to design mutants that would be resistant to the inhibitors. So the work I will highlight right now is the work of the group of Dr. Christopher Léger from Marseille in France, and they developed a method called protein film voltammetry that allows one to measure kinetic rates for the binding and unbinding of the inhibitors CO and O2. And on top of that, what they did was to design different mutants. You can see here, in this row, you have the wild type, and the different rows are different mutants, where they tried to block or hinder the access of the inhibitor CO and O to the catalytic site. So first, their hypothesis was that the main bottleneck was composed of the residues in position 74 and 122. Then they started introducing mutations in these two positions to see if they could hinder the passage of this inhibitor to the catalytic site. And here we have the kinetic rates measured by them, especially for the dissociation of CO and O2. And we can see here that first they did not obtain mutant enzymes that were resistant to the inhibitors, but what they could obtain was enzymes that had a slower passage for the inhibitor CO and O2. And then apart from this, there were other computational groups also working with this particular hydrogenase to investigate what would be the main bottleneck for inhibitor binding and unbinding. So from the previous work from Christopher Léger, for instance, from this paper, they hypothesized that the main barrier was between residues 74 and 122. But other computational groups also suggested that actually the main barrier was between residues 74 and 476. And this is something that we also were interested in testing in our simulations. So for the first part that I'll present right now to you, that was published in JCIM last year, what we wanted to do was to investigate the pathways for CO1 binding from the nickel ion hydrogenase from the sulfonylurea. And also from using these results, we wanted to propose new mutations to possibly turn the nickel ion hydrogenase resistant to inhibitors. And last remark I want to do for you is that in the experiments from Christopher Léger, in his kinetic rates, we have the events highlighted here in green, where we have the formation of a covalent bond between the catalytic sites of the enzyme and CO. Then we have the rupture of these bonds, and then CO diffuses away to the solvent. In our simulations, because we are using conventional force fields, we do not see the step of bond rupture, but we start here when CO no longer has a covalent bond with the catalytic site. So we simulate the step where CO is in the catalytic site and we see diffusion away to the solvent. And this is, according to the experiments, the main barrier for the dissociation, because the reaction in the catalytic site for the formation of the covalent bonds is faster than the diffusion of the gas inside the enzyme. And then as methods, what we use here were basically molecular dynamic simulations. So first, we use the Newton equation of motion to propagate the motion of the atoms in the system. So A is the acceleration over the atom, F is the force over the atom, and M is the mass of the atom. And then from this equation, what we need now is how to calculate the force over the atom, and we calculate the force by looking at the interaction energy of one atom with all the other atoms in the system. And this interaction energy is given by the bonded and the non-bonded interactions. The bonded interactions are given by the bonds, angles, and torsions between the angles. So we have bonds between two atoms, which are given by this equation, a harmonic potential. We have angles between three atoms, which are also modeled by a harmonic potential, as you can see here in this graph. And we have the dihedrals, which are modeled by a slightly different equation. Dihedrals happen between the planes formed between four atoms. And for non-bonded interactions, we basically have the Lennard-Jones equation to model van der Waals interactions. And we also have the electrostatic interactions to model interactions between charges. And with all of this, and depending on the size of the system, we can achieve timescales that go through from nanoseconds up to microseconds of simulations. And this still poses a challenge to us to investigate protein ligand dissociation like we want to do here, because experimentally unbinding can take milliseconds or more time to happen, while our simulations reach the microseconds time scale at best. So here we can see that there is a challenge to bridge this difference in time scale. So what we do is to use what we call in the literature enhanced sampling methods. So there are a lot of computational strategies and enhanced sampling methods to facilitate the observation of unbinding events. And the method I will highlight for you today is tau-RANDI, which was developed by my former postdoc supervisor, Professor Rebecca Wades from the Heidelberg Institute of Theoretical Studies here in Germany. And if you want to know more, you can check this paper, but I'll provide a brief explanation here. So the idea of tau-RANDI is that we facilitate ligand dissociation by applying a force of constant magnitude and random orientation in the center of mass of the ligand. So first, we need a model or the experimental structure of the protein ligand complex, and then we equilibrate the temperature and the pressure in the system, and then we start applying a force in the ligand here in pink. And then after time delta t, we check the displacement delta x of the ligand. So if the displacement is larger than a threshold, then we keep applying this force in the same orientation. But if the displacement was smaller than the threshold, then we change the orientation of the force randomly. And we keep doing these steps of propagating the motion of the atoms in the system and checking the ligand displacements until we have the event that we want to achieve, that in this case is protein ligand unbinding. And I want to make two extra remarks here. So first, note that we are applying a force over the system, but we are not making any corrections for the force application. So this means that we don't have the absolute residence times, but we have relative residence times. And the relative residence times, they can be compared, for instance, among different ligands binding to the same protein or different mutant proteins binding to the same ligand. And the second thing is that we don't do one simulation, but we do tens or hundreds of simulations to have a good average for our relative residence time for one particular system. All right, and then I like tau-RANDI especially because it's simple and it has very low computational cost. So if we have a few hundreds of nanoseconds, we can already estimate relative residence times across different ligands. And this is a figure from a previous review that I wrote about enhanced sampling methods. My lab recently also wrote another review, so you can check these two reviews if you want to know more about enhanced sampling methods. And here, basically what we have is the experimental residence time for different system and the simulation time required to predict absolute or relative residence times. And here, every group of points or every point represents a different computational method. And we can see we have a lot of variations of metadynamics. We have tau-RANDI, we have target molecular dynamics, also developed here in Germany. And what I want to highlight here is that tau-RANDI, you can see, is the second fastest method by the time this review was published. So meaning that it really requires not a lot of computational time to already make predictions. And Now going to the results, so we applied tau range to that all the mutants that were present in that table that I presented to you from Christopher Léger. So here we have experimental residence times, and here the computed residence times from tau range simulations for the Y type and 10 different mutants of the nickel iron hydrogenase. And again, the experimental results come from that paper that I highlighted in the introduction from Christopher Léger. And here we are looking at CO dissociation, and we apply the dissociation force of 1 kilocal per mole angstrom. So before this work, me and everybody else in the literature had applied tau range for protein drug complexes. So our work, this particular work, was the first one to adjust tau range to small gas-like molecules. And this particular work was also the first one to simulate in a comprehensive way all the mutants with kinetic rates available for CO dissociation. So here we have a coefficient of correlation of 0.79 and a Spearman ranking correlation coefficients of 0.75, meaning that tau range can accurately rank relative residence times in agreement with the experiments. And here we have one example of a pathway for CO dissociation. So here's the catalytic site. CO is here in the start of the simulation, and CO moves, and then it's out of the enzyme in the end of the simulation. And okay, and now that we know that our simulations reproduce the experiments, then we moved on to a lot of data analysis. We were particularly interested here in the pathways. So here, first thing we did was to identify different tunnels in the crystal structure for possible ligand unbinding. So here we use KVerdoc and this particular PDB, and we have 9 different tunnels that are numbered here for T1, T2 up to T9. And then next, what we did was to analyze the unbinding pathways in the simulations using a tool called Aqueduct. So here we analyzed the unbinding pathways with aqueducts, and then we matched the pathways with the tunnels identified in the crystal structure. So here what we have is the statistics of tunnel utilization for the Y type and all the 10 different mutants. And these are the results from 75 independent simulations for every panel that you see here. And here the enzymes are ordered from shortest up to longest residence time. So here shortest residence time, and here from shortest up to the longest residence time. And this is the shortest and the longest residence time. And then first of all, what we see for the Y-type enzyme, here we have the statistical utilizations of pathways T1 up to T9. We see that the preferred pathways here are basically pathways T1 and T2. So this pathway here and this pathway here. And I want to highlight to you that also these two pathways are related. If you look at this picture here, we see that the beginning of these pathways, these two pathways, they are pretty much close to each other, right? Right here. So they start as the same pathway and they bifurcate in the end around this alpha helix here. And then, so for the Y type, the preferred pathways are pathways T1 and T2. And when we look from shortest to longest residence time, we see that still the preferred pathways are all the same pathways T1 and T2. But for longer residence times, like this mutant and this mutant, we see an increase in alternative pathways like pathways T7 and T8, as you can see here and here. So we barely see utilization of pathways T7 and T8 in the Y-type enzyme, for instance. Okay, and then one of the most exciting results from my perspective is actually when we look at the Y-type hydrogenase and the pathway utilization for the different gas molecules. So in the previous slides, I was showing pathways for different mutants and for CO unbinding, and here we are only looking at the Y-type enzyme and now different ligands, CO, O2, which are the inhibitors, and A2, which is the substrate of this enzyme. So here we can see that for the 3 different gas molecules, the primary pathways, the most populated or the preferred pathways are still pathways T1 and T2, T1 and T2. But when we look at the alternative pathways, if pathways T1 and T2 were not available, we can see that molecular hydrogen can use different alternative pathways, for instance, pathways T9 or T4. So hydrogen, the substrate, has a higher chance of using the alternative pathways T4 and T9, which are not used by the other gas molecules like O2 and CO. So based on this, what we propose in this study is that to obtain hydrogenases that can be resistant to CO102 is basically to design enzyme mutants which block pathways T1 and T2, and at the same time open the pathways which are the preferred pathways for molecular hydrogen, like pathways T4 and T9. And then for the second work that I want to present to you today, this was published in JPC Letters this year. What we wanted to do is to propose a data set to benchmark pathways probabilities from enhanced sampling methods. So one of the challenges is that it's very difficult to obtain binding and unbinding events from unbiased molecular dynamic simulations to have a data set for a benchmark. But in this particular case, we were able to obtain hydrogen binding and unbinding events to two different hydrogenases, the one I presented before and the one from megalodesulfovibrogigas, to use it as a benchmark. And the idea here is that if we have these binding and unbinding events from unbiased MD simulations, we can use these methods to benchmark enhanced sampling methods to see if our enhanced sampling methods are not somehow creating changes or biases in the pathway probabilities that would be expected or observed in unbiased simulations. And this time, so for this particular part, we used regular conventional molecular dynamic simulations, and we didn't use any enhanced sampling methods, and we basically observed spontaneous binding and unbinding events for molecular hydrogen. So we had a total of almost 20 microseconds of simulations for each of the two hydrogenases. And for the first hydrogenase, the DF hydrogenase, we had around 40 binding and unbinding events, and we are able to compute a K-on value of 9.5 10 to the power of 7 molar minus 1 second minus 1, which is close to the experimental K-on value available in the literature. For the other hydrogenase, the MDG hydrogenase, we had around 100 binding and unbinding events. So again, once we saw that the simulations reproduced the experimental data available, we went on to data analysis, and we again identified the unbinding pathways from these simulations, and we computed the percentage of utilization of these pathways. And one very interesting thing that we saw, so we basically first of all compared the binding and unbinding pathways of hydrogen for the DF hydrogenase and for the MDG hydrogenase. So pathway symmetry is expected for equilibrium systems according to the principle of detailed balance or microscopic reversibility proposed by Boltzmann. And we can see in this two system that there is pathway symmetry, or in other words, the pathway probabilities for ligand binding and ligand unbinding are the same in these two different enzymes. So basically, we applied a chi-square test to see if there were statistical differences, and there were no significant statistical differences. Which to me is a very nice, exciting result from the physical perspective. And then what we also did was to compare the pathway probabilities for unbinding from the unbiased simulations with tau range simulations. Again, results for the DF hydrogenase and for the MDG hydrogenase. And what we saw is from our chi-square test is that for the DF hydrogenase, there's no significant difference between tau range and the unbiased simulations, which is good. But for MDG, there's a significant statistical difference. And this is mostly coming due to differences in utilizations of the pathways T2 and T5. So we can see that T5 has a higher chance of being utilized in tau range, while this is not so high in the unbiased simulations, while T2 has a much higher probability of utilization in the unbiased simulations. So we are still trying to understand why tau range doesn't exactly reproduce the unbiased simulations for this enzyme, but for the other enzyme, everything was as expected, and tau range has the same pathway probabilities as the unbiased MD simulations. And then the other thing that I wanted to show to you today, remember, were the two proposals for the different bottlenecks for ligand binding and unbinding. And I told you there were two different proposals in the literature. So what we did to settle down the matter was basically to compare the experimental residence time with the distance between the proposed bottleneck residues in our simulations. So here, for the first bottleneck proposed was between residues 74 and 122. So here we have the logarithm of the experimental residence time, and here the distance, the minimum distance between these two residues from our tau range simulations. And what we see is a strong and negative correlation, which is an indication that the distance between the two residues really is a bottleneck and modulates the residence time. So for instance, for the Y-type enzyme in our tau range simulations, we observed a large distance and also a short residence time. So in the Y-type, there's a large distance, then there is no bottleneck. But in these two particular mutants, for instance, we have now a shorter distance and also a long residence time, meaning that these two residues are blocking the passage of the inhibitor. And now for the second bottleneck, the distance between the residues 74 or 426, when we compare the experimental residence times with the distance between these two residues, what we see is a strong but a positive correlation. This is an indication that this is not a real bottleneck, because for instance, when we have a long residence time, this means that the passage is hindered. We also have a long distance, meaning that these two residues are not acting as a bottleneck here. And then what we saw with our unbiased MD simulations, so we did some Markov state modeling using PyEMMA, and you can see the results of the Markov state modeling here. So basically what we saw is that we have two states for this bottleneck. So here we have two progress coordinates identified by time-independent component analysis. And here we have the free energy as obtained from the Markov state modeling. And we can see that we have two main states for the distances between the two bottlenecks. So here we have the open state represented here with this distance of 8.3 angstrom between the leucine and the valine. And here we have the second state, which is the closed state, which is the states which you observed in experimental crystal structures, which has a shorter distance between the residues involved in the bottleneck. So another thing that we propose now in this paper is that we could also pay attention to this bottleneck and modulate propose mutations that modulate the behavior of this bottleneck and put it mostly in the closed states to further block the passage of inhibitors to access the catalytic site. All right, and then wrapping things up, this is again that graph that I showed you before between experimental and computed residence times. And here I want to highlight that there goes a lot of effort for simulations and also data analysis. So here we have 11 different enzyme forms, and every point here represents the average and standard deviation from 75 independent trajectories or 75 different unbinding events. So in the end, to determine unbinding pathways, we had to analyze more than 800 trajectories. And we used Aqueduct, which is a very useful tool, but it does require some manual matching between the unbinding pathways between different mutants and also between the mutants and the tunnels in the crystal structures. So something that we wanted to do here was also to facilitate and streamline our data analysis. So what we did was also developed, we also here developed Path and Hydro, which is basically a set of machine learning models to identify unbinding pathways and trajectories in an automated way using a supervised machine learning model. And if you want to know more, the paper was published this year at JCIM, and the code is also available on GitHub. So basically, for this particular part, we used machine learning and we tested different classical algorithms for classification, like logistic regression, support vector machines, and the algorithms that had the best performance and that we used in the paper were algorithms based on decision trees. And this is one example just to explain to you how decision trees work. This is a decision tree to predict solubility using two features, the log p value of a molecule and the molecular weight. So basically, while we are training the decision tree, we choose the one question in a node to separate the data set in two. So here, the first question that was decided for this particular decision tree is whether the log p value is larger or smaller than 3.11. So if the log p value is shorter than this, then we go to this branch, otherwise we go to this branch. And then this next question to ask in this node is the value of the molecular weight and whether the molecular weight is larger or smaller than 268 grams per mole. So if it's smaller, then we go to this branch. Otherwise, we go to this branch and then we take a decision about this particular small molecule. For instance, whether this is soluble in water or not. And this is one example of one decision tree. So nowadays we don't train a model based on a decision tree alone, but we rather use ensembles of decision trees. So what is very common in the literature and that we used in this particular paper, in one case, a random forest, which is an ensemble of decision trees trained simultaneously or in parallel, but with slight differences in the training set between every tree. And another common method is adaptive boosting, where we have an ensemble of decision trees that this time is built sequentially, where the next tree is built to correct the errors of the previous trees. Are you still there, people? I hope you are.
Yeah, we
Yep.
are here. I actually, I think we have some more people joining also. So yeah,
Okay, thank you. And then now going a little bit into, well, if I'm blowing up the time, Edward, feel free to cut me off. I may have a lot of slides, I don't know. But anyway, I'll move on, Edward. If it's too much, cut me off. All right, so now going to a bit more detail. So this was a basic explanation of the decision trees. Now going to a bit more detail about the data set. So our data set were the molecular dynamic simulations and the pathway attributions we did before using the previous program called Aqueduct to identify the unbinding pathways in the trajectories. And we built basically two models, one for binary classification and one for multi-class classification. The model for binary classification classifies the pathways between primary and secondary, and primary pathways are the pathways with higher probability and that are structurally related, basically pathways T1, T2, and T7. And secondary pathways are all the other pathways. And you can see here the training set made of the DF hydrogenase, CO, and hydrogen, the validation set, and the test set made of O2, which is another molecule not included in the training set, and made of the MDG hydrogenase bound to CO, H2, and O2. Note that the MDG hydrogenase was not included in the training set as well. And you can see that there we have a large problem of class imbalance, meaning that we have much more instances of one class, the primary pathway, in comparison to the other. And we also had a second model that did multi-class classification. So in the multi-class classification, the model basically identify the specific pathway T1 up to T9. And then the data set basically, again, was made of our trajectories obtained from tau Randy. And for the training set, again, we have a CO and H2 dissociation from the DF hydrogenase. And the test set was CO, H2, and O2 dissociation from the F hydrogenase and from the MDG hydrogenase. And as features, we used the fraction of time of ligand residue contacts during our simulations, and 804 features because we have 804 residues in the hydrogenase. And the algorithm, as I told you before, we tested different classical algorithms. What worked best for our results for the validation set was adaptive boosting for the binary model and random forest for the multi-class model. And here again, if you see, now we have the distribution for the multi-class model, and we have again a problem of class imbalance because we have much more instances from pathways C1 and T2 in comparison to the other pathways. So we did oversampling here to increase the number of instances of the less represented classes. And then here are the results for the binary model and the multi-class model. So to follow the quality of our models and how good the predictions were, we followed statistical metrics that work good for imbalanced data sets. So here BA is balanced accuracy and MCC is the matches correlation coefficient. And the maximum value they can have is 1. So the closest to 1, the better the prediction is. So here we have the results for the training set, the validation set, and the test set. And we can see that for the training set, the metrics work very well. They are all close to 1, which is kind of expected. What I want to highlight is the test sets, particularly parts of the test sets that were not included in the training set. So here we have the test set for the DF hydrogenase and O2, which was not part of the training set. We can see that the model works okay-ish. The MCC is not great, but okay. And the model works particularly well for a new hydrogenase, the MDG hydrogenase. So here we have the MDG hydrogenase and different gas molecules, CO, H2, and O2, and the model works very well for this new hydrogenase, meaning that the model is transferable for other small molecules and other hydrogenases not present in the training set. And here is another common metric, which is a ROC curve, which is a curve of the false positive rate versus the true positive rate. And here we have the AUCs, the area under the ROC curve, and the closest it is to 1, the better. And we can see that the areas here are usually pretty decent, all above 0.7. And then, if you are not super tired, I can talk a little bit about toll-like receptors. Do I still have time to talk a little bit about toll-like receptors, Edward? What do you think?
Yeah, not bad. It's raining here, but we can have some 5 minutes. If the electricity doesn't disappoint, we can have 5 minutes, please.
Okay, I'll wrap this up in 5 minutes, otherwise you cut me off. Okay. So very quickly now an application in drug design. So toll-like receptors are parts of the immune system and they are involved in different diseases. And what we wanted to do here is to investigate pathways for agonists and antagonists and binding for toll-like receptors. So this is the structure of the toll-like receptor. This is a homodimer and this is the binding site. So this is one part and this is another part of the dimer. And here we have the agonist. Here we have 2 cationic antagonists with a positive charge here. This is important information, remember, of this positive charge. And here 3 neutral antagonists. And then we did tau range simulations and we obtained 100 independent unbinding events. And here we have the statistics. So what we observed is 2 different unbinding pathways, the external pathway here in red and the internal pathway in blue. And what is very exciting is that there is very clear pathway preference. So the agonist only unbinds through the external pathway and the cationic antagonists only use the internal pathway, as you can see here. And now the neutral antagonists, they don't have pathway preferences, as you can see. They can either use the external or the internal pathway, as you can see in these 3 graphs here. And why is it the case? It's all in the details, in the interactions. So when we look at the bound structure of the cationic antagonist, we see that the antagonist forms a ionic interaction with the glutamate 427, and because of this ionic interaction, unbinding to the internal pathway is greatly facilitated in the cationic antagonists. Now for the neutral antagonists, what happens is that there is no ionic interaction. So this glutamate forms a hydrogen bond with tyrosine and blocks the unbinding pathway. So in the neutral antagonists, internal and external pathways are equally difficult, so there's no pathway preference for the neutral antagonists. And that's it. So I hope I convinced you that computational methods can be used not only to predict kinetic rates, but also to provide nice mechanistic insights about protein ligand unbinding. Here we saw spontaneous binding of hydrogen to nickel iron hydrogenases, and we proposed this as a data set to benchmark enhanced sampling methods. I also discussed with you what are the pathway populations for the inhibitors and for the substrate of hydrogenase, and this offers us opportunities to design inhibitor-resistant hydrogenases. And finally, I presented to you PathFinHydro, which is a group of machine learning models to facilitate the identification of unbinding pathways in nickel iron hydrogenases. And that's it. So thank you so much for your attention. Most of this work was made by my fantastic PhD student Farzin, this person here, and the machine learning part also had help from my postdoc Iris, this person. And the last part, the toll-like receptors, were done by Valerie, this person here, and it was also performed in collaboration with Professor Gerhard Woba from the Free University of Berlin. And thank you for your attention.
Thank you very much. Thank you. We can begin to ask questions. Let's have 15 minutes of question time from the audience.
Questions, people?
Those of us participating online, please, you can unmute yourself and then ask your question. Okay, I think I'm going to ask a few questions if there are no questions, but please feel free to unmute and interrupt me while I ask my questions, because I am not the most important for the question part, but I think we will all learn from the questions I may ask. So my first question has to do with the transition from what you observed from the literature review and how you etched out your research objectives. How did that, did you have to do the review yourself, or you just got the inspiration from someone's review? That's my first question. And my second question has to do with the force fields for simulating metal protein complexes, and how handy does it get? Is it available everywhere, or you wrote these force fields yourself and probably do some benchmarking to see the accuracy of these force fields before proceeding with the molecular dynamics simulation?
Oh, okay. So that's, I'll start with the second question. So that's a fantastic question. I didn't comment too much about the force field, but honestly, developing force fields, so in principle, force fields and parameters for metal enzymes are usually not available in the standard version of the force field. So a person usually spends a PhD to parameterize a metal center because it's a lot of drama, it's a lot of work. The particular parameters are used in this work. They were these parameters were available in the literature before because other people parameterized and tested these metal centers. But that said, if you want to work on this, this would be great because this is a great limitation of working with metalloenzymes nowadays. So one of the questions we got, especially for the machine learning part, one of the reviewers wanted us to test other hydrogenases, other enzymes, and one limitation we had is that there were no metal parameters available, so we had to test enzymes that had parameters available for the metal centers. So I think this would be terrific if someone had the time and wanted to put the effort to parameterize more metal centers, because this is something missing in the literature now. So the study of hydrogenases is limited by the metal centers with parameters available. So there are more hydrogenases with different metal centers, but we cannot do simulations because we don't have the parameters. And quantum mechanical calculations to parameterize then is not my business and not my expertise. So yeah, if one of you want to do it, I will be very happy. So, and the first question was about the review, right?
Yeah. How do you translate from the review paper into getting the research idea before? Yeah.
So yeah, so the review, so basically, why did we write this review? So the first review, my boss Rebecca was invited, and invited me to write. So an invitation that is not really an invitation, but okay. Then I wrote the review, right? So basically, I think it's nice to write reviews because it gives you an overview of the field and what is done, and especially what is missing. So when you sit down and read a few papers of the last 2 or 3 years, then you have a very clear view of what people have accomplished and what is missing. So I think it's very nice to write reviews to see very clearly, oh, this is what people have achieved so far, and this is what people are not talking about. So it's a mix of reading a lot to see what's in the literature, and then brainstorming to see, well, what else could be done that I don't see in the current papers. I think it's a wonderful exercise to come up with new ideas for research. I really like writing reviews.
Wow. Thank you very much. We have one question in the chat, but I like to give the floor to Wyclef or Mansour to ask directly. Please, can you unmute yourself to ask?
You can read his comment on the chat. Yeah.
So the first question says, how do you decide that XYZ is the relevant descriptor to address my problem? And also the second says, how does machine learning based descriptors perform when compared to physics based descriptors for your case?
Okay. Okay. The question's here.
Yeah.
A lot of difficult questions about machine learning. So how do you decide that XYZ is relevant? That's a great question. Yeah, so that's the $1 million question. But yeah, I think in the end of the day, when we are dealing with chemical problems, we have to think about the chemistry, right? That's the thing. So yeah, first, Wycliffe, I hope I'm pronouncing your name correctly. Thank you for the question. So yeah, I'm currently working in predicting kinetic rates, looking at features of protein ligand complexes as well, apart from the work I presented here. So how do you decide if about your first question? We have to think about what's relevant for the problem, right? So particularly for if we want to predict properties that are related to protein ligand complexes, then we certainly need to think about things related to the protein and the ligand. So yeah, that's how I would approach the problem. If we have a data set in chemistry, we have to think about the chemistry and what is important to make predictions for that particular data sets. And at the same time, we want descriptors that are informative, but also that are simple, because if it's too difficult to obtain such descriptors, for instance, if we need experiments that take as much time as the property you want to predict, so then you just do the experiment instead of trying to predict something, right? So we really have to come up with something that is simple and meaningful for machine learning. This is question one, and now question two. How does machine learning based descriptors perform compared to physics based descriptors for your case? So yeah, for my particular case, what I did was basically use the descriptors that I had from the simulations, because basically I want to predict something that is happening in the simulations. I did not test machine learning based descriptors, and I don't think this would work for this particular case. Now, broadly speaking, about kinetic rates, I think when we want to predict, for instance, kinetic rates, I'm currently only using machine learning based descriptors, let's say, because now obtaining physics based descriptors, I think this would be a bit more expensive. So yeah, I hope I answered your questions. And now we have also another question from Damilari Babatunde. Yeah, nice to see you again.
Yeah, please go ahead.
Hello, can you hear me?
Hi.
Thank you, Prof. In one of the, thank you for this is a very good presentation. I really learned a lot. But in one of the slides, I don't know where you have the training set and the test set. Yeah, no, no, yes. Is it this? Yes, the data set. You said the trajectories, I say that the training set is different from the test set. I thought in machine learning, we take about 80% for training set and 20% of the same data set. But I see that in the test set, we have the CO, H2, and oxygen um, added to the test set. So I was kind of confused in this part. Is it an applied machine learning or is it still the same data set?
Okay, so that's a relevant question. So yeah, in principle, what we do is to pick up one single training set, that in this case, one single full data set, for instance, in this particular case, the data set for CO and H2, and split it in training and test set. So that's what we have here, for instance, here and here. So these are the same groups of simulations, let's say. And then what we had here, and that is somewhat that people can do in machine learning, is to see if we can transfer or extend this same model to other test sets that are less related to the training set. So for instance, and that's why we have these different test sets. So this first test set is very related to the training set because the enzyme and the small molecules, they are the same. And this is the standard way of doing things, I would say. So pick up one single data set and split it in training and test set. Like you said, 80% of the data in the training and 20% in the test. And here we took an additional step to see if our model would be transferable to other small molecules like O2 and also other enzymes that are like more different and that were not present in the training set. So basically we did an extension of our test set to test the transferability to different conditions, let's say. I hope it makes sense. Did I answer your question?
Yes, thank you. It's transfer learning. You mentioned it. I understand. Thank you.
Okay, you're welcome. More questions, people? Ask me anything.
Well, I think I have advantage. Please, I want to ask something more related to biotech and probably very naive, but I ask anyway. So, where do you have somewhere in the literature where we have an array of inhibitors for this nickel ion hydrogenases, or we only have carbon monoxide and oxygen as the main known inhibitors of the activity of this enzyme?
It's another inhibitor related to nitrogen. I don't remember specifically which molecule, but there's one extra inhibitor, yeah. But then I don't think there's a database or anything. We have to dig the literature to search for this. But then one of the problems is that there are not a lot of kinetic rates available to benchmark simulations. That's very difficult. That paper I showed in the introduction is the only paper with kinetic rates for inhibitors, unfortunately. And then if you don't have experimental data, it's hard to make benchmarks to see that your simulations reproduce the experiment. So yeah. But
Okay.
yeah, there's one extra inhibitor and I don't remember which particular molecule this is.
Okay. Okay. Right. So apart from smaller molecules like carbon monoxide and oxygen, if we want to look at smaller gases that have a similar molecular volume like these ones, and then predict whether they inhibit or do not inhibit. In this sort of thinking, do you simulate in vacuum, or you simulate in the presence of water to have your observables? I'm just getting curious with this.
Oh, okay. Yeah, so one thing that I didn't discuss much here is yeah, the inhibition. So one of the things, one of the reasons why, so the main reason why they are inhibitors, O2 and CO are inhibitors because they can, first, they can reach the catalytic sites, and second, because they can react with the catalytic sites and form a covalent bond and disrupt electron transfer there. So I think the best thing, if you want to test in silico, if they are inhibitors, is first to test if the small molecule can react with the catalytic sites, and second, to test if they can reach the catalytic sites. And then, at least for the parts of whether the small molecule can reach the catalytic sites, we did our simulations in solvent, and ideally the simulations should be done in solvent. Yeah, that's where the enzyme exists, I would say. So simulations, yeah, here should be in solvent. But then if you want to check if a small molecule is really an inhibitor, we really would need quantum mechanical calculations here to check whether covalent bonds can be formed with the catalytic site.
Great. Another question in the chat says, from Sabino Redumensa says, what is the difference between validation and test sets?
Yeah, good question. So yeah, that's the number one source of confusion in my master students in my group because it is really confusing. So basically, when we set up when we have our complete data sets, it's common to divide it in training, validation, and test. So training is what we use to train the model, and validation is what we use to fine-tune the hyperparameters of the model. And I didn't discuss this here, but hyperparameters are basically the parameters of the model itself that need to be optimized. So for random forest, for instance, we have the maximum depth of the tree as one hyperparameter, the number of trees of the model, the number of features we considered at every node to make a split between the nodes. So, and to refine this, so basically to refine the hyperparameters of the model, we use the validation set. And the test set is what we use to check how the model would behave in a real case scenario with unknown data. I hope it's clear.
Yeah, he says it's clear. Thank you. Okay, last one will be to ask in this validation space where you have to do parameterization, how expensive is this computationally? Do you need a supercomputer, or you can just do this on your laptop?
These particular models, so this data set, you see, it's not so large. I think we had roughly 1,000 data points. And we use classical algorithms like random forest and adaptive boosting. So we did this without a high performance computer. So you can do it on your laptop. You can train and use the method on your own laptop. But when you move to neural networks, for instance, then sometimes, depending on the size of your data set, it's useful to move to high performance computing.
Great. All right. Thank you very much.
You're welcome.
There is one new. What form does the feature data takes? What data type is used to represent the trajectories that represent a pathway?
Okay, so basically, the feature is basically the fraction of time of a residue ligand contact during the simulation. So basically, it's a number that varies from 0 to 1. 1 meaning that the contact is there 100% of the simulation. 0, the contact doesn't appear in the simulation. And then yeah, that's how we represent the pathway. Don't have exactly the pathway, but we have rather the fraction of time that the ligand residue contact is there for every residue in the protein. And that's our feature set, and that's how we represent the trajectory there in the machine learning model.
He's asking if you could repeat the explanation again.
Okay, I didn't see. Okay, no problem. All right, I can repeat the explanation. So basically, let me see the question. Okay, what form the feature takes and what data type is used to represent the trajectory. So basically the feature is what we use to represent the trajectory. So the feature is the fraction of time of the ligand residue contacts in the trajectory. So for every residue from 1 up to 804, we have one number that varies from 0 to 1. 1 means that the contact was present to 100% of the frames in the trajectory, and 0 means that the contact was not present in the trajectory. So basically that's how we represent the trajectory by these features, which are again the fraction of time of ligand residue contacts during the simulation. I hope it's clear now.
Yeah, he says it's clear. Thank you very much. I think we have drained so much out of you, and most of us have learned a lot. Personally, I have really enjoyed your talk, and I'm sure the audience have also done the same. And at this point, we would like to, on behalf of the entire institute and our audience, say thank you very much for your time and for this beautiful presentation.